Here's the puzzle. In the earliest stages of development, the embryos of closely related species tend to look quite different, very divergent. But as development proceeds, these different species often tend to converge on a common form in the middle of development. And then as development continues to the adult stage, the forms tend to diverge. So we get a lot of diversity early among related species, conservation in the middle, and diversity later. And this forms an hourglass shape and is known as the evolutionary hourglass. And it's one of the great unsolved puzzles in evolutionary biology. Vertebrates follow this pattern. Vertebrate embryos are quite diverse early in development. In the middle stage of development, vertebrates tend to converge and share neural tube, the somites or segments, and the pharyngeal arches, which are the primitive structures that develop into the head and neck. But as development continues, the adult forms then tend to look quite different. So once again, we get this broad, narrow, broad hourglass shape. Molecular data support this hourglass process or shape in other groups of organisms as well, where we see in the earliest stages of development, the gene expression patterns between different species are quite different early. The middle stage of development shows a conservation and convergence in patterns of gene expression, and later stages of development diverge. So again, we have the hourglass pattern. And so again, the puzzle is, why is the middle stage so conserved? And there are a variety of explanations, but none of them are particularly compelling. And so what I want to do today is suggest that the conservation in that middle stage in development arises because in the middle of development is when it's necessary for embryos to translate between different types of information. And that domain translation is necessarily conserved. So let's start with an example, the morphogen gradient, which is one of the most important and conserved aspects of development. Early in embryogenesis, the cells divide, and so you've got a bunch of cells among the early embryo. And then one of the first grade patterning steps, often, is that a chemical gradient arises from one end of the embryo to the other. And that chemical gradient then signals that one end is going to be the head, and the other end is going to be the tail. And so you've got the linear axis through the embryo that's made. And this is a translation between the geometry of the embryo and the spatial location of each cell and the gene expression pattern. So it's a translation from geometry to gene expression, or space to gene expression. And the morphogen gradient is highly conserved across different species. So that suggests that one of the reasons for pinching in evolutionary conservation is this kind of domain translation. We'll come back to the morphogen gradient in a minute, but first let's just back up and talk about the logic of domain translation a bit more. Domain translation is a fundamental and common feature of essentially all engineered systems. Engineered systems often have to translate between different types of information, and they typically do so through conserved protocols. Protocols are just simply a technical term for translation between different domains in this context. So let's talk about an example, computers. In computers we have software, and software does a variety of things. It's very diverse. But essentially all software operates in the same way. When the software is working on some data and wants to store the data, it tells the operating system, okay, here are some data, hold on to these data, I'm going to need them later. And the operating system then takes that message in the data and talks to the hardware and tells the hardware, physically we need to store these data, so you do this and physically put these data away. And then later the software says, okay, give me those data back, tells the operating system that. And the operating system tells the hardware, okay, give me the data, and the operating system passes the data to the software. So all of the communication between software and hardware happens through the conserved protocols that are the core of the operating system. So what we end up with is very diverse software that can do all sorts of things, and very diverse hardware that is built in a variety of ways to make computers. But the protocols in the core or the kernel of the operating system tend to be highly conserved, because that's what allows talking between the domains. And that conserved operating system kernel is basically set on the common Unix or Linux core that's common to essentially all modern computers. So we see tight conservation in the middle of two diverse components. So that's the notion of conserved domain translators. Going back to development, let's go back to the morphogen gradient then. And the theory predicts that these translators in biology will have three properties. First, a domain translator compresses information. So in a morphogen gradient, we have a variety of different geometries that compress down to a single linear signal. That's the domain translator. It's a low-dimensional signal. If you hold that low-dimensional signal constant, the downstream pattern remains the same, even if you perturb the upstream pattern or change the upstream pattern. So it's only that low-dimensional signal that determines the response downstream. So we get great compression of different geometric patterns into a low-dimensional signal. This is a common feature then, compression. The second great feature of a domain translator is evolutionary conservation, and that's what concerns us the most. These translators tend to be conserved because they're allowing communication between two different domains through a particular interface that's the translator. In order to change the translator, typically requires changing both domains and the translator in simultaneous ways that prevent perturbation, and that's hard to do. And so the domain translators, once they form, tend to be highly constrained and conserved. We see that in morphogen gradients. The chemical gradient that can pattern Drosophila embryos can pattern a frog embryo, and the chemical gradient in a frog embryo can pattern a Drosophila embryo. So we see conservation over vast evolutionary distances for these morphogen gradients. And the third property is fragility. Because the domain translator sits between two crucial aspects of development, if you perturb a domain translator, you often cause significant perturbation in the pattern that's formed through development. Morphogen gradients are fragile. If you perturb the bicoid chemical that's involved in Drosophila embryos, you get widespread anterior disruption of the developing embryo. In humans, if you perturb sonic hedgehog, which is a key morphogen gradient component, you get widespread defects in the head and face. So we have compression, conservation, and fragility as common properties of these translators. Now in vertebrates, we have morphogen gradients, sets the head to tail axis, and then the next great step in patterning is the formation of the segments or somites, the physical boundaries between the segments. And the way that happens in vertebrates is through a conserved segmentation clock. Segmentation clock works by having an oscillating, traveling wave of a chemical reaction go down the embryo axis. Oscillation is a temporal fluctuation, and the segmentation clock times those fluctuations to set the physical boundaries of the somites or segments. This is a translation from time to space. It's highly conserved, it compresses information, and it's fragile. If you perturb it, you cause widespread perturbation. Then, the next step in embryogenesis is we have the somites or segments, and those have to be patterned into particular tissue types to do things. And that's the job of the Hox gene cluster. And Hox genes then translate spatial location and developmental history into the fine-grained tissue gene expression patterns. So translation of history and space into gene expression. Hox genes are compressed information, they're conserved, and they're fragile. And so we see that vertebrate embryogenesis is a sequence of multiple domain translations, or protocols, we can call them, forming what we could call a protocol stack. Morphogen gradient sets the axis, segmentation clock sets the physical location of the segments, and the Hox genes pattern the segments. This notion of protocol stacks is fundamental in engineering for complex control systems. The way in which data get passed through the internet follows a TCP/IP protocol stack. These protocols tend to be conserved because once a protocol is set, it's important that they remain conserved to form these domain translators. So my argument is that development is a sequence of domain translations that form a protocol stack. The protocols tend to be conserved and they're in the middle of development. And so that pinches the middle of development in terms of evolutionary conservation and explains the wide, narrow, wide hourglass pattern that's commonly observed. Thank you.